Alright, what we're going to do today is we're going to conclude Chapter 7 just to summarize what we've been talking about for the past week, almost 2 weeks really. And then we'll get into Chapter 8, which I did provide for you the handout. I'll give you just a couple pages to take notes on for the Chapter 8 introduction. And then you'll have access to the electronic files after the weekend. You can reprint or whatever you want with those. Chapter 8 and 9. Chapter 8 will take us about 2 days to get through, and then Chapter 9 will take us it should take us a day to get through if we do okay with Chapter 8 material. It's going to deal with cell replication, looking into different types of cells, whether you're looking at a cell of the body or a cell that's considered to be a sex cell or a gamete cell. And so where I'm going is I'm going to ask you to read Chapters 8 and Chapter 9 over the weekend. Take notes so that when we get into class, it'll be a ton easier. And that Learn Smart, you can't get into the textbook. The Learn Smart is a great way to just kind of passively kind of get ready for the lecture, so that'll help you. And always, if you're in a pinch for time before Tuesday, at least read the Chapter 8, Chapter 9 summaries at the back of the chapters. Okay, so at minimum, read those. But I would ask that you go into detail so that the lecture goes a whole lot easier. So just to summarize really briefly, transcription, translation. These two processes are involved with what? RNA is definitely the molecule that's an intermediate molecule. And what's the purpose? What's going on with transcription and translation? They're making a protein. Making a protein. And basically everything that a cell needs, not everything, but a high proportion of the molecules in a cell are proteins. We've named them specifically like proteins. We talked about ATP synthase, or we talked about the enzymes that regulate the glycolytic pathway. We've even talked about proteins that serve as receptors, right? So protein is just a super generic word to describe an amino acid built something. But all of the amino acid built somethings, these proteins, get their origination from these processes called transcription and translation. And sometimes these processes are collectively just oversimplified and called gene expression. Why might transcription and translation collectively be summarized in this term called gene expression? Why is that a good term, an acceptable term?
Because it's how they're shown and how they function.
Very good. It's shown. It's how a gene is shown, and also is going to apply its function. If a gene is expressed, that means it's been transcribed and translated into a working what? Protein. Okay? Very good. Where does the process of transcription occur? In a eukaryotic cell's nucleus. Okay, so we're going to focus on eukaryotic organisms. And it produces for us different types of what molecules? RNA molecules. We learned that the RNA molecules are formed, what's the word in relationship to the DNA strand? Complementarily. So if there's an adenine DNA nucleotide, and if I make it an RNA nucleotide, the RNA nucleotide would be a uracil. Or if there's a thymine DNA nucleotide, there would be what type of RNA nucleotide? Adenine. Okay? It just goes in complement nature. Most of our RNA molecules are how many strands? One or two? One. Okay. Everything we've studied so far is one strand. We kind of had a discussion about it a week ago. Okay. Very good. The RNA molecule that we kind of focus on is which? Which molecule that kind of gets the highlight? The accolades. When we follow it up with translation. mRNA. Why mRNA? It's a messenger. It has the codes. And we'll read it in sequence of 3 called what? Codons. Okay, very good. What does the mRNA associate with in order to carry out translation? What does it have to first bind with? Even before then, it has to bind with a ribosome. First a ribosome. The mRNA has to bind with a ribosome. Does that make sense? was said? I didn't hear. I want to make sure you're okay. What did you say? Did you?
I was going to say anticodon, but you're saying the general protein.
Yes. It will. Yes. The mRNA will bind with tRNA anticodon, but first it has to bind with the ribosome. But you're correct. The mRNA does bind with the tRNA anticodons, which also have what type of relationship? Complementary relationship. Very good. Not only does the tRNA have anticodons to bind with a messenger RNA, it also has what? A specific amino acid that it's going to bring in order. The ribosome, the messenger RNA, and the transfer RNAs will all work together to bring or to build a what? A protein. Okay. Any questions so far? I should have in the bottom of your Chapter 7 handouts kind of like a summary. Is there a summary table for the translation on the bottom of Chapter 7? You can turn to it. I want to point some things out for you that you might not have thought about. You probably don't, up to now, have thought about DNA, transcription, RNA, translate, protein, right? So sometimes the common question is, well, then what happens with the mRNA? What happens to the ribosome, subunit, and all these things? Anybody thought about that? No. It's okay. But it's an important question to think about if we're trying to synthesize protein. What happens to an mRNA? Well, the mRNA can actually be retranslated. Okay? If it's still structurally sound, if it still has this 5' end correct, and this 3' end is correct, and the middle is all correct, if it's still structurally sound, it can be retranslated to make another what? To make another round of protein. And the same protein will be coded for. So transcription can occur once, but I might have dozens or hundreds of proteins actually translated by using one little mRNA transcript. Once translation terminates, at some point the messenger RNA needs to get degraded because we don't need to just indefinitely make this protein over and over. Okay? If you remember some of the terms like negative feedback, at some point we'll have enough protein, stop the translational process, and that mRNA will actually be degraded or broken down, chopped up, cleaved. Not right away. The duration of mRNA may last a few minutes, maybe upwards of like 30 minutes, maybe an hour or something like that, but not collectively a long time in the span of a cell's life. It's just a quick process. What questions do you have at this point about the roles, the location, the function of transcription or translation? I'm going to encourage you to keep doing the practice of the complement between the DNA and the RNA to make the protein. Okay? If we have time, we'll go back and practice some more. But I really want to get into cell division in Chapter 8. Any burning questions before we get into Chapter 8?
On the homework, when you had to do the translating the DNA to the RNA, it asked me again for letters that aren't a part of the bases. They're not UAC. Are we going to need to know that for the test? The amino acids?
No. You will need to know how to read that table that I provided for you guys in your handouts. You'll need to know how to read this. I will provide this, but I'm certainly not going to ask you to know the codon sequence for all the amino acids. I don't even ask that of my biology students. Only people going to junior and senior level biochemistry, then they need to know that. But if you know what an amino acid is, I'm happy. You know how it gets coded for, I'm even more happy. Additional questions? extras of the same,
Yeah.
I should. At the end of class, I can give you some.
Okay.
But I don't have any. You can also make up your own sequences.
Yeah, that's what I've been doing.
Okay. But I'll check. I should have some extras with me.
Okay.
Here's an animation. I've not watched it in a while, so I'm not sure on how thorough it is, but it at least kind of re-summarized transcription and translation for us. Transcription, we noted in the eukaryotic cells, does occur inside of the nucleus. And so this image, this is actually zooming in microscopically at the nucleus. This isn't an entire cell, this is just the nucleus, which I can presume because I see the pores and I can see the double membrane. So it tells me that's the nucleus, not an entire cell. Transcription can occur during a process, during a phrase that we'll use next week during interphase. So basically when a cell's living, surviving, doing its thing. It's not actively dividing, replicating the entire cell itself. During transcription, I'd like for you to, and I'm sure we mentioned it, but I just want to re-mention it. Transcription is just copying one gene. It's just one tiny portion of the DNA. It's not the entire genome being replicated. It's just one little segment that goes for one protein. Okay, so it's very specific. It's a very regulated process. And what we noted during transcription is there's an enzyme called, what kind of polymerase is it? What kind of polymerase? RNA polymerase. So it's a polymerase. Polymerase is just kind of a generic word. It's like it makes a polymer. Well, it's making RNA. So we call this RNA polymerase. That's active during transcription because we're making RNA. In this case, we can actually see where the DNA double helix has already been separated. It's, as you might imply, it's way more involved than just magically the RNA polymerase knowing where to go. There's other proteins, but that's beyond where I expect you to go. But I'd like you to understand that in order, once a gene is ready to be transcribed, RNA polymerase is available, separates the double helix, breaking what type of bonds? Hydrogen bonds between our double strands to expose the DNA template. What color is the DNA strands, backbones colored? The sugar, the phosphate is colored which? Of the DNA molecule, it's colored the red. The red is the DNA. The reason we can tell is we look forward a little. Okay, so this darker colored is the DNA strand. The lighter colored yellow strand is our RNA, and it does bind complementarily. The RNA is built in which direction? Which end is put on first? 5' end, and then it ends with the 3' end. The phrases are initiation, then what? Elongation, then termination. At the end of termination, or at termination, we can see that RNA nucleotide cleaved off, and now it's free. What I didn't mention, but it's certainly in your textbook, and therefore I won't hold you accountable for it if you didn't cover it, but I mentioned that the RNA molecules are processed. Okay, they're not just magic. They're not ready to go just directly from the DNA template. The ends have to be modified. The middle might have to be kind of chopped up or something like that. The tRNA has to fold. The ribosome. And so there's additional modifications. But if we follow a modified messenger RNA, it exits the nucleus through what type of structures? The pores of the nucleus. Goes out into the cell's cytoplasm. And it's not like a snake. There's proteins that actually pull it, right, and move it out there. But looks happy. But and now that the RNA template is out in the cytoplasm, it can interact with ribosomes and then transfer RNA molecules to carry out the next process of what? Translation. During translation, we said it's going to occur in the cytoplasm or where? Where could it also occur in the eukaryote? Well, yeah, definitely in the mitochondria, but in the rough ER. Okay? So the mitochondria in the chloroplast actually do this too, but I didn't mention that. But for a protein that's going to be secreted rough ER, or a protein that's going to work inside of the cell, it's a cytoplasm. As long as you acknowledge it's not where? Not in the nucleus. Okay. What is the purple thing? Ribosome. The purple thing is the ribosome, and it does have enzymatic properties, so you could say that, but most specifically it's a ribosome. The yellow thing is the what? Which RNA? Messenger RNA. And the orange things are the tRNA, the transfer RNA. In this image, it's really oversimplified. You can really easily, hopefully, easily see the anticodons. They look like prongs on a fork. They're nucleotides. They bind complementarily to the messenger RNA. What are the tic-tac looking things up there? Amino acids. They're not tic-tacs, they're amino acids. And eventually these amino acids will be bound together through what type of bond? Peptide bond. Peptide bond. The peptide bond is the name of the covalent bond that binds our individual amino acids. We can't see a bond here yet because they're not adjacent to one another. The assembly, the translational machinery will assemble. You can kind of see the large ribosomal subunit complex. As I mentioned as well on Tuesday, this is just a cross-section. This would all be internalized inside of this three-dimensional globular protein. All right. The dark green and the light blue stretchy things in between the amino acids, that's representing the peptide bond. So I'm going to go back and show that I kind of simplified it. But these, can you see where these bonds are trying to be represented by this animation? Notice that once the first amino acid, which is which amino acid? MET, methionine. Notice that once it forms its peptide bond with the second amino acid, what happens to its transfer RNA? It's released. It can go pick up a new what? Amino acid. And if it binds, it could bind back in. And it just goes in sequence. Okay, so here we're in the middle, we've already initiated, and now we're in the middle of the elongation process. Each amino acid is joined with the subsequent amino acid via a peptide bond. And they just move through, reading codon by codon. Can you see how it's going in segments? At some point, there'll be a stop codon reached where there's no new transfer RNAs binding, so the machinery is disassembled, and we have now released our, and we call it a protein at this point. And then, depending on the stability of this messenger RNA, a new machinery could reassemble. Does that help a little bit? Now, make a note also following translation. That protein is the end point, right? We're kind of like a protein. But if you tie into chapter, I think, 2 or 3, the proteins then have to fold and all kinds of stuff. So also the protein might have to undergo modification to get its ultimate structure, its three-dimensional structure. All right. Chapter 7 discussed a pretty intricate process of transcription and translation, which produced the RNA complement in the nucleus so that we can manufacture proteins in the cell cytoplasm. Okay? It's very specific for specific genes. However, where we're headed in chapter 8 and 9 is we can actually, cells can copy the entire genome and not just specific little sequences. Okay? And so, if we look at the entire genome being replicated, that will happen when a cell gets ready for cell division. So, this is what you're looking for a moment ago. This will be the Chapter 8 handout for any class. This is what you'll take notes on. You pass these over. Here's you.
Yeah.
These will be provided for you electronically later after the weekend. In Chapter 8, we're getting ready for a process called cell division. Cells have to divide. You have an idea about this, right? You know, even for biology, and your parents told you about the birds and the bees, or you heard about the bees, cell division, replication. You start from one cell, end up with trillions of cells. You think about something complex about like a human. Okay? So cells must divide. And when they do so, they've got to replicate all of their DNA accurately and quickly. Okay, so that's what we're going to address here in Chapter 8. We're going to talk about the DNA replication process in order to produce identical cells. So in Chapter 8, we're making cells that are identical. Maybe you want to circle that, highlight it. We're making identical cells. During this process, this one type of cell division. We'll talk a little bit about, not really the whole human life cycle, but we'll look at some different types of cells in the human body. And we'll get into more detail for DNA replication and introduce this term called the semi-conservative process. I don't think we'll get there today, but if we do, we'll talk about how DNA actually assembles into a chromosome, and that'll set us up for the actual cell division itself. And you've probably heard of it, mitosis. You already talked about it in lab, right? Maybe? Got yeses and nos. So maybe do it. You might have. I don't recall either if y'all been through it yet or not. But when cells get ready to divide, right, non-sex cells get ready to divide, the process is called mitosis. And so we'll go through each of those phases. There's something called a concept map. You should have something similar to the end of your chapter. And so we're going to first just talk about DNA replication today. We're going to talk about the enzymes that are involved to regulate the replication process. And then when we come back to class on Tuesday, we'll talk about just this term called the cell cycle. And that's also the handout you'll turn in for credit next week. And then we'll look at the phases of the actual cell division, which is called mitosis. Just wanted to remind you, going back to the very beginning of the semester, we're basing the foundation of this class on this scientific theory called the cell theory. And if we looked at the three tenets, we said the cell is the basic unit of life, which we covered in Unit 1. And we also identified that all living things are comprised of different cells. And so that limited to us to just cells, not viruses and tables and whatever. Okay? So anything that's considered to be living is built of cells. And a third important tenet that was added a few years later, after the foundation of the theory, is that all cells must come from pre-existing cells. So in other words, life just doesn't spontaneously arise. Okay? And the way that this happens is through different types of cell division. It should be figure 8.1 in your textbook, illustrating, for example, the human life cycle. And right, you fundamentally know, right, to make a human, sperm and egg, right? Those are two separate cells that actually have half as much DNA as necessary to make a functioning human. And so therefore those two cells have to fuse together during a process called fertilization. Post-fertilization, the embryo is formed, or described here called a zygote. And the embryo, human embryo, and most embryos just kind of start out as just, almost look like soccer balls. They're just kind of boring looking blobs, undifferentiated little cells. And so those cells will grow and they will divide and they will divide, and eventually they'll begin to specialize into, say, cells that make your brain, or cells that make your heart, or your muscles, or whatever. And at some point, a baby will be born and then be an adult, okay, which themselves could reproduce. Okay, so chapters 8 and 9 will tie together this reproductive process. Not all living organisms have this exact same life cycle as humans do. Okay, so if you think about the insects that we'll be seeing in the ground soon, now that the weather is better, you'll see those nasty little grubs, and there'll be nasty little beetles, right? And so creates little different life cycles, or plants have different life cycles. And so, but this is, we can understand the human life cycle because we are human. Okay, so we know a little bit about the human body. In chapter 7, we're going to study this process called mitosis, which is illustrated for just growth and development, not non-sexual reproduction, or non-gameted reproduction. Chapter 9 will highlight the purple area during the meiotic cell division. It's a really cool image of human fetal development. This image on the top left, where it says 1 day, is just an egg, a female oocyte. Looks pretty, I mean, it almost looks like a textbook is like a circle. I mean, it literally is a round sphere, undifferentiated. Huge cytoplasm. And then on the outer periphery, can you see it almost looks like something, you see the little stuff kind of hanging off of it? In that mix will be cells that came from the woman's ovary, okay, so the cells kind of protect it, but also there would be sperm cells trying to bind and attach. After that fertilization process, can you see how this one cell is divided into 2, and then 2 would divide into 4, and then 4 divides into 8, and it just keeps going and going until we kind of get this really cool called a morula, like a soccer ball structure that I identified. And then it goes on to develop, it's called a blastocyst. You can kind of see the big kind of hole over there. And what's interesting, I think we showed an image early in the semester, these human embryonic development looks identical to cattle embryonic development, which is identical to human, which looks identical to a lizard, which is identical to a snake. We all start out kind of pretty well similar, like little blocks. Yeah. So it's pretty interesting, or at least it is to me. Hopefully it is to you as well. And then at some point, cells will begin to differentiate into forming, say, the human placenta and the other cells that form the brain, and it just, right? So we start out from something very generic looking into something that's very well formed and specialized. I learned on the radio yesterday that some scientists were actually able to map the little fetal brain. They're actually, up till now, the human fetus brain development, difficult to study because technology and moral ethical issues. But some scientists were able to get some fetus. You know where they come from. So they were able to get some fetuses and study their brain development. Now we have a map, and so we're better able to understand fetal brain development, which we care about this because we can be able to diagnose brain diseases early in development. And so it was interesting, the scientists who were doing the work were saying actually they believe some cases of like autism spectrum or schizophrenia, you know, typical brain problems or diseases actually are occurring here in the fetal stage, not in like youth, right, or adolescent groups. So it's actually early on, before someone's actually born. As one grows, let me back up. So we don't look like day one. Okay, so we don't continue to look like a little round ball. Some cells have to grow and some cells have to stop growing so that we can get this molded look to ourselves, as illustrated in figure 8.3 in your textbook. This process, this regulated process for cell division is called apoptosis. Okay, sometimes you read it apoptosis. I had an instructor when I was in graduate school, like apoptosis, so I just, apoptosis it is, and not apoptosis. You sound like a redneck, so apoptosis. So if your friends talk about apoptosis, you would correct them. Don't sound like a redneck. No. The process of apoptosis is just, I've got a spot for you to define it in your handout. It's described as regulated cell death, or sometimes it's described as programmed cell death. Apoptosis is just regulated or programmed cell death. 2, right, is the purpose of, say, development. To form appendages and tissues, etc., right, for differentiation. Okay, so there's a purpose for the cell death. We can also see apoptosis important for kind of killing cells that might potentially be cancers if not killed. So apoptosis is not only important during early growth and development, it's also important for preventing unregulated cell division, or what we call cancer. So it's just it's an internal, it's pre-programmed. I mean, it's way more than I understand, but it's a pre-programmed role in development that helps your fingers separate from one another, your toes, your nose, whatever, you name it. It gives us our look. And we would just continue on into big blobs, round, squishy blobs.
And wouldn't that lead to abnormal cell division as far as
Yes, if apoptosis doesn't happen after development, that's what we would call as cancer, as unregulated, very much so. Before differentiation, some tissue would just be extremely large. Apoptosis certainly has its role. When you first hear the word like cell death, it sounds bad and lethal, but it has an important role for development and cancer kind of protection, if you will. The role of cell death and cell growth, or the relationship, the ratio of cell division, or I have here cell growth and cell death, must be in balance. In a mature person, cell death and cell growth should be matched. If they're not matched, what happens? One takes over the other. So if there's more apoptosis, the whole organism will eventually die. If there's more cell growth, squishy blobs or cancer. Okay? Very good. So in the developed adult, the ratio of cell division and cell death should be matched, so they don't have more loss or gain of tissue. Just have some examples here of why we might need cell division. Okay, so I already listed some as well. So just growth, like a person's growth from the fetal to a young person to an adult. Plant growth, kelp growth in the ocean, coral growth, right? Tree growth, whatever. Right? In order for something to grow, it needs to undergo mitotic cell division. Differentiation, development. Another statement I have on here is like wound healing. Okay? If you have a sore, you don't want that sore to just persist indefinitely throughout your life, right? You want the cells around it to divide and reproduce. So wound healing is an important example of why mitotic cell division is necessary, besides just growth and development. Looks like I have a typo. It says some cells will divide continuously. Okay, some cells will divide continuously. So, for example, some cells do divide continuously, like the cells in your bone marrow. So throughout your life, your red marrow continually divides to make for you what? Your what? The bone marrow, the cells in bone marrow divide continuously to make for you your blood cells. So some cells retain the ability to continuously divide. Your skin cells, they certainly get replicated all the time, just sitting here taking notes or just sitting on your chair, you're sloughing off, you're losing epithelial cells, so we need to replace those, right, continuously. However, in our bodies, some cells lack the ability to continue to divide after a certain point of development. And most notably, these include cells of your brain and your spinal cord and your heart and your skeletal muscles that you use for movement. They lack the ability to divide after a certain point in growth and development. Which, if you think about it logically, if I cut my skin, the cells around it will divide and replace it. If I cut my spinal cord, are those cells going to naturally just divide? No. If I have a traumatic brain injury from an accident or military duty or something like that, those cells, they're not going to divide without some type of medical intervention. We're getting to where we can change that, but they're not programmed to just divide. So some cells do divide, some cells lack the ability to divide. When we look at cell division, what has to happen is the entire genome has to be replicated. What does that mean, the entire genome? What's the genome? We defined it, I think, a week ago. The genetic material found inside of an organism, everything. It's the entire genetic material. So when a cell divides, not just a couple of its genes, not just the ones that it's going to express, but the genes, all of the genes, both the ones that make useful proteins or the ones that aren't even used. Remember last week we said not all proteins are actually expressed. Okay? And so prior to cell division, if we look at body cell division, the entire genome has to be replicated. So we're getting more broad than just copying a little gene. And so we've got a large task. And if you remember the millions of base pairs, that little graph I showed last week, how many millions of base pairs there are in, say, just a human, it's got to be quick and it's got to be accurate. If it's not accurate, there might be what? The question you guys had about we're going to be responsible for mutations. And if there's mutations, it could be bad. Okay? And so right before a cell divides, it has to copy its entire genome accurately and precisely. At the end, we'll see there are two identical cells, and they're called daughter cells. That's just a term. It doesn't describe gender to the cells. It's just saying it's the one that was made following cell division. So a daughter cell is a term that you can use if you're a male in your skin cells that divide. Okay, it's a daughter cell. It's the one that was made. But the two daughter cells are genetically identical because their genome, the original cells, the parent cells, DNA was accurately, precisely, quickly replicated. The process for DNA replication is going to be important for you to remember the complement nature and the structure of the DNA because we're still here. Okay, so we're still just studying DNA. When a cell is activated to divide, whether it be through growth and development or wound healing or whatever the case may be, the entire genome is replicated, and it's replicated in a complement fashion. So we still have the complement fashion, but we need to copy the entire genome. And so what happens is, well, let's review. DNA has how many strands? 2 strands. If we want to read them, we have to separate them, split them. Okay? And in DNA replication, not just one, but both strands are actually copied. Okay? So that when each strand is copied, we have 1, 2 full sets of the genetic material. Okay? When the 2 strands are copied, half of the strand goes to one cell and the other half goes to the daughter cell. This term is highlighted here. It's called semi-conservative. Semi-conservative. What that means is when DNA is replicated in a semi-conservative fashion, the 2 strands are separated, And each strand then is copied and then goes to a separate cell. So the daughter DNA is half old, half what? New. Okay. So I'll try to repeat it in the same fashion. So during DNA replication, right, the entire genome is replicated. Each chromosome will be separated and copied so that each strand serves as a template, and when it's copied, they'll separate. So that now the double DNA of the double strands, of the two strands, half has old, if you will, parent DNA. No, I'm not going to use the word parent. I'm going to take that word back. Old DNA, original DNA. And the other half has the newly synthesized DNA. Each strand has original, as each DNA molecule, one strand is the original DNA, and one strand is the new complement. I'm sorry. Let's take one strand of DNA, separate it. Let me rephrase this word strand. Take one molecule of DNA, which is comprised of two strands. We separate the strands so that the nucleotides are exposed. Each is copied. That was pretty awesome. Sorry. So that, okay, the new copies have original and new DNA. I think that actually might work. Does that actually work? The original DNA are my pointer fingers, and they're separated and then copied so that the new strands of DNA have new and old DNA in them. The people in the online class are like, what is she doing? Okay, so did that phenomenal visual help a little bit? Make sure you use the right fingers, you don't get in trouble. So instead of doing this, I could say semi-conservative. DNA replication, entire genome replication, is a semi-conservative process. What that means is each new DNA strand is comprised of new and old, newly synthesized and old original DNA. So in this image, the original DNA, or what's going to be called the template DNA, the sugar phosphate backbone is colored a which ribbon represents the original template DNA? The dark blue. Okay, very good. The enzymes are not shown in this image. We'll get there in just a minute, but it is separated and it is going to be copied so that the new complement of DNA, sugar phosphate backbone, is colored which color in this image? Kind of the light gray white area. And if we look a little bit closer, we can see a new strand, 1, 2. Semi-conservative, because in these two daughter strands, there's original and now new DNA in the molecule. These will eventually fully separate. And then we can get on to when we get on Tuesday. Where does it go? So if I use the word semi-conservative, does that make a little more sense than say 5 minutes ago? In order to do this, there are certainly going to be enzymes to regulate this process. And I have a note for you in your handout that it does occur in the eukaryotic cell's nucleus. It does occur in the cell's nucleus. And then we'll address that a little more. In order to replicate the DNA accurately, precisely, in a semi-conservative process, it's enzymatically regulated. We know that enzymes work quickly and efficiently. Some important enzymes and other molecules involved in this process include an enzyme called helicase. There's also some things called binding proteins, primase, DNA polymerase. Now don't get this confused with what? RNA polymerase. RNA polymerase is used in which process? Transcription. DNA polymerase is used during DNA polymerase is used during replication of the entire genome. Alright, so we have helicase, some proteins that stabilize, one called primase, DNA polymerase, and another one called ligase. Save that one for the last because sometimes that's a little bit more confusing, but hopefully less confusing after today. Alright, so helicase is an enzyme. Anything that has the suffix ASE here is an enzyme. Okay? So helicase, probably hela, acts on the helix. Okay? And what it does is it helps unwind that helix. Remember, our helix, it helps unwind it. Is that me or somebody else? Not me. Alright. So helicase is an enzyme that binds to a DNA molecule that is ready to be replicated, and it just relaxes that helix. Because we want to get in it. It relaxes the helix, and it also separates the strand. So it's not only relaxing, it's breaking what? The hydrogen bonds. In this image, and also from I've used these images for your handouts that you guys will turn in next week, but that purple blob. It's a protein, it's an enzyme, and its job is to bind to the DNA molecule. As well, you can see these images are shown behind the DNA, but they would actually wrap around and touch a three-dimensional interaction with the DNA. But can you at least see where the DNA is separated? Now keep in mind the anti-parallel nature of the DNA. Don't forget that. The two strands are anti-parallel, so we'll go there in just a minute. But right now we're just using helicase. What does it do? Loosens up and unwinds and separates, breaks those hydrogen bonds, separates the DNA molecule into two separate strands. That DNA inherently wants to be in a double helix. Okay, those hydrogen bonds are attractive. It wants to bind to one another. But in order to copy the DNA, those hydrogen bonds have to be destabilized. Okay? And so helicase will break the bonds, and there will be additional proteins that will come into the machinery and they prevent those strands from actually temporarily forming those hydrogen bonds. And these are called the binding proteins. So the little binding proteins described here stabilize the strands. They keep the strands from rejoining, reannealing is the term, one another. The little binding proteins in this image, they're also shown kind of superficial, but the little tiny little blobs are representing the binding proteins. Keep the strands separated.
Like holding them back.
Yes, like holding them back, because they are electrically wanting to attract. Yes. No, not yet. So we separate the strands, we hold them back, and now we can keep those nucleotides exposed so that we can do what with them? Copy them. Okay, make them complement. Okay. Now look, in transcription, we just use one DNA template. In this case, we're going to copy both. Okay, so we'll have multiple enzymes working at the same time. First thing is, there's an enzyme called Rna primase. Do you have this on your list? I just have it listed as primase. You can put in there parentheses Rna primase. Primase, enzyme, what does that word prime mean? Besides the mathematical operation that we've used before, prime. Ever prime something? Start it, right? It starts it. Okay. So to get the process, the site going, there's a little initiator RNA sequence is called RNA primer, and it's added by an enzyme called RNA primer. The RNA molecules are represented in the green strands here. They still bind complementarily, but the RNA nucleotides, they'll eventually get cleaved off, but they're saying they get everything primed, ready to go. The enzyme RNA primase binds those there. Okay? These are little RNA primase. Once the polymerization has been initiated or started, then our enzyme called DNA polymerase can get involved. DNA polymerase is an enzyme that does what? It binds nucleotides. What kind of nucleotides? Well, it does it complementarily. What type of molecules are we making? DNA, right? So it takes DNA nucleotides and puts them in order in a complement fashion based on this template DNA. It will join adjacent to the little primer region. So we have our RNA primer in green, and then we can start the replication. We can see the nucleotides in blue, the DNA nucleotides in blue. This way we know which strand to basically copy or when to adhere to. Okay. It is reading the DNA backwards so that the new strands are built forwards. Okay. That's correct. DNA polymerase reads the template strand backwards. Or what does that mean backwards? From which direction? 3 to 5 prime. It reads it 3 to 5 prime so that the new strand is built 5 to 3. Very good. So DNA polymerase reads the original template which direction? 3 to 5 backwards, so that the resulting daughter strand is 5 to 3. Okay. Now, here's where it gets tricky. Here's the tricky part of DNA replication. Is the original parent strands are anti-parallel, and they both get created at the same time. So one can go continuous, the other one can't go continuous because its sequence is all twisted up. The one strand, we'll call the lagging strand, is made in sections as the DNA unwinds. And so let me show it here. If you look at this DNA helix, let me restate this. All of the DNA is not magically separated at the same time. Tiny little sections of DNA go through this process. So it's not like all the millions of base pairs are like separated and waiting. Okay? It's just little sections to regulate it. Okay? Because the two strands, when they separate, they do run anti-parallel to one another. The DNA polymerase can only read what's exposed. It can't read where helicase has not opened up. Okay? And so if you look at this image on the right, we can see our double helix on the far left. You see how it's still twisted in the helix? Right, so helicase is not worked that way. And in this case, it's showing the way that they've designed it. But the helicase is going to work from this image from the right side of the page to the left side of the page, or from the 3' end to the 5' end. Okay? On this top strand. On the other strand, it's being read 3' to 5'. So notice on this 3' to 5', the rest of the 3' is still wound up. Can you see that? So DNA polymerase can still work, but it only works a short segment at a time. Eventually, helicase will open up, and a new strand will open. So then the polymerase can bind, but then we'll have a little segment. The original template can just open. Since it opens, it just polymerizes.
does the new strands, do the same thing as the old? Or they stay separate? Do they?
They will stay separate. The daughter strands at the end, yes, they will inherently begin to fold once they start doing some stuff. So before I move on, I want you to at least kind of superficially acknowledge, because it's anti-parallel, the way the helicase opens, one strand can just go. And the helicase opens, and the daughter strand can change. But the other strand has to kind of catch up. It works in segments, one into the other. And if I think, I'll try to find an animation for you. I've forgotten what it is. This image figure should be 8.5. Will help visualize what I was trying to illustrate for you. On the top, we have what's called the leading strand, or continuous strand, where the original strand is open 3 to 5, and it's just easy to go. Conversely, on the lower hand side, so when you see this daughter template is just continuously synthesized 5' to 3'. The other strand has to work in segments. It's called the lagging strand. When those little, on that lagging strand, because it starts and ends, there's going to be sections where nucleotides aren't joined, so we need to join them. What enzyme can join those little segments? Ligase. Ligase joins. And in this image, the little rectangular looking thing is ligase. And it's going to go through and join the fragments. And they're called the Okazaki fragments. You can just call them fragments. That's how I have a hard word to become familiar with.
So it joins fragments and they're just lagging behind.
Joining segments made from the lagging strand.
So like the separate strands that are made are called
The separate strands are technically still just the daughter DNA.
okay, I'm confused with the
Let me show you an animation in just a minute to help hopefully unconfuse you. But before I move on to the animation, I'll see if there's additional questions. I'll hopefully address your question with the animation. It'll help. I hope. Any other questions before we move on to show the animation? Yes?
So it's the strand that works from 3 to 5 is the lagging one, and the one that works from 5 to 3 is the is that every time or not every time?
It's always, it depends on because the DNA is a three-dimensional molecule, DNA polymerase always reads the DNA 3 to 5. So it just depends on where you are in the direction of the way that thing is moving. And so it just depends on the DNA strand. In this case, they have the DNA strand so that it's ordered 5', 3' like we would read it. It is a three-dimensional molecule. Okay?
So it's not always just perfectly lined up.
No, it is not always at all.
Alright.
I would say never actually like pretty like this or simple like this. But where I can hopefully better address your question is because because DNA polymerase always reads the DNA, so the darker blue, from the 3' to the 5' direction. Okay, so it's working in this image from the right side of the page to the left side of the page on the top original strand. Okay? Because that strand is 5' is being read, R-E-A-D, from 3' to 5', and the machinery is moving I don't know how to say other than
With
left.
the flow.
With the flow to the left of this page, okay? Its next template should be always available. Okay, so it doesn't have any hoops or hurdles, it's just continuous, it sufficiently works. The other strand is also read in in a 3' to 5' fashion. However, the original 3'.
I understand.
Okay, it's there. It's just not exposed, and so the DNA polymerase can only work with what's exposed. And so it'll work 3'. They'll make this. This will open up a new zip, and it'll be 3'.
And it's
I get
not
that.
exposed because it's red packed.
It's not exposed yet because the DNA polymerase does read the original strand packet.
Okay.
Additional questions? This animation is pretty simple, but hopefully it'll help a little bit. Or a lot. Prior to cell division, we said the entire genome needs to be replicated. We're going to look at just a single little segment of a DNA sequence, right? It's not specific. The sugar phosphate backbone's been oversimplified with the red ribbon. The nucleotides also been oversimplified with the the green and the blue rectangles. Okay. An enzyme that separates, and again, it's not magic like this, but you can see the little arrow, right? What is that arrow representing? Helicase. It's going in there, breaking up that helix, and separating the hydrogen bonds. At this point, the nucleotides are exposed. Binding proteins will stabilize it. I do not believe they're shown in this image. It's just showing they're magically separated. Okay. Now, DNA polymerase, well, first RNA polymerase will prime the reaction. And then DNA polymerase can kind of join the party and read each strand, the DNA strand, what we considered so far backwards, 3' to 5' directionality. Where the question was, well then, because the two original strands are anti-parallel, one strand can be read continuously, and the other strand has to be read in segments. The DNA polymerase is always trying to catch up. And it's going to print out little short segments of copy. In this case, which strand would be the continuous strand? The top or the bottom? The bottom is the continuous strand, and the top we'll see is the lagging strand. Now we'll see another molecule. Now that we have on the far right, we can see another round of 3', 5' directionality exposed, and we can now copy. But those little segments have to then be joined, and the enzyme that joins them is called what? Ligase.
And that purple thing that was moving was that the DNA.
The purple thing in that image was representing DNA polymerase. What I want you to add to your notes at the bottom of this is how this happens. It doesn't start at the very beginning and go straight in order. It doesn't do all the DNA at once. There are millions and millions of base pairs to copy. What there's many DNA polymerase, many helicase, many ligase molecules available that are activated during this process. So that on a given chromosome, bubbles, they're called replication bubbles, are generated. So that tells us little sections of a single strand are actually activated at the same time. And eventually, the individual bubbles would be joined by an enzyme called ligase. Yes. When an entire genome is replicated, it doesn't occur magically, spontaneously, and it doesn't go like in single file order. Many enzymes available, lots of DNA to work on. So what happens is the replication occurs at, they're called replication bubbles. So they kind of start in the middle, anywhere on a chromosome. At some point, each replication bubble will form with a new replication bubble, and that would be through the use of ligase. And at some point, the entire chromosome has been copied, And the two strands would do what? Separate. I'm pretty confident this animation shows the replication bubble and formation or fusing. So if I could have your attention and look at, okay, this would be a single replication bubble. And if you look at it on a large scale, right, so you can see, hold on. In this case, we can see 1, 2, 3 replication bubbles. And eventually the enzymes, the multiple enzymes would work together until they fuse. And now we have 2 full sets of DNA. Those 2 strands have half the original and half of the newly synthesized DNA in them. Does that help address the question about first the fragments and then 2 the replication bubble? Rachel? The fragments, did you see how the one has to kind of wait? Do I need to go back and show that animation again, that section? I'm not okay with think, so we want to be sure we see that. All right, so in this section, we have one strand, which is the continuous strand. It's not labeled 5', 3', or whatever, but I can see that the bottom strand, the DNA strand, is the 3 to 5 because of the way the RNA polymerase is working. The other strand is 5 to 3, so DNA polymerase has to start at the 3' end, go to where it worked, and then jump back. Kind of like leapfrog or something. I don't know, it has to go back. Right? It kind of goes to the end and it goes back to the beginning.
So the modules, the new ones, the lagging strand.
The newly formed, yeah, the copy of the lagging strand. Those fragments. Right? When that DNA polymerase goes back, it meets those 2 strands have to be made as one. So watch this animation. On the top, DNA polymerase is reading 3 to 5. And now, a new DNA polymerase combined to this 3' end, right, close to the 4th. And so on the top strand, you can see how it's going to go. It reaches where something's already been made, and now it can go back and a new molecule can go back and new molecule DNA polymerase can now work 3 to 5. So there's more 3' end exposed. I would like for you to add a note about DNA polymerase. It's a pretty amazing enzyme, how fast it works, and how efficient it is, and how accurate it is. Think about you, for example, when we were doing our transcription, I think I had like maybe 12 nucleotides, and out of the 12, you tried to erase 3 or 4 of them, right? So you had about 75% accuracy. DNA polymerase has to copy millions, plural, of DNA nucleotides within a few minutes. Okay? And so it's really accurate, it's really fast, it's really precise. Not only does it polymerize the molecules, but what I want you to add is it proofreads its work. And if there is an error, if it detects an error, then it will fix it and then rejoin. What we're adding is DNA polymerase also proofreads the newly synthesized strand to check for accuracy. This collectively that we just walked through is a process called what? Replication. It occurs prior to cell division. It occurs prior to cell division. DNA replication copies an entire genome. It copies everything. Whether it has been used or will be used or will not be used or has never been, whatever. If it's there in the nucleus, it's going to get copied prior to cell division during this process called replication. I'll conclude our discussion here today. As I mentioned at the start of class, it's imperative that you read the rest of Chapter 8. I would certainly read this front matter by tomorrow so you don't lose the ground that you gained today. And then on Tuesday, we'll talk collectively in one day. On Tuesday, we'll talk about mitosis, which is your handout number 9. And then on Thursday, we'll talk about meiosis. So I'll probably give you a handout on Tuesday to turn in on Thursday. So, yeah. Okay. All right, so we'll see you guys in class on Thursday. These weeks go by too quick. We'll see you later.